So today's lecture will be given by Professor Yoshihiro Taguchi from Faculty of Science at Chuo University. And I would like to briefly introduce his background. So Professor Taguchi received his PhD at Tokyo Institute of Technology and worked as an assistant professor in the same university. So when I was an undergraduate student, I was one of the students who took his class of the thermodynamics. Okay, and so Professor Taguchi became an assistant professor in the Faculty of Science at Chuo University, and then now he is a professor there. And he originally worked on the nonlinear physics, but now he works on the bioinformatics using technology in the physics. Okay, and he has many interesting papers and also books. So, and today he will give a lecture titled "Application of Tensor Decomposition Based Unsupervised Feature Extraction to Genome Science." Okay, now then, Professor Taguchi, please start. Thank you.
Oh, thank you very much. Oh, nice introduction. So, and I would like to thank the people here to come and to join my lecture. And if you have any questions during my lecture, you can interrupt me and ask me some questions. And it is also applicable for the people who are attending this lecture via WebEx. So I'm not sure how the people who are attending this lecture through WebEx can ask me. Maybe Professor Sakumura can notice if someone in WebEx would like to ask something to me by chat. Please let me know. Okay. Then the people who are attending this lecture through WebEx can chat and ask Professor Sakumura to ask me any questions. So I'm not sure the people here is interested in my talk because my talk is a little bit special. So I would like to know the background, the people here. So he said the people here mainly from biology and mixture of biology and informatics. So who came from biology? No? Okay, so you. And please raise a hand in WebEx people who came from biology. How many? Let me know, Professor Sakumura. So how many people? Please raise a hand. Yeah. How many? And so mainly people, how many people came from informatics? Okay, so how about the people in WebEx? Okay, so many majorities came from informatics. Okay, so this is what I have expected, so no problem. So today I would like to discuss these topics. So I'm applying the tensor decomposition to biology, especially in genomic science. And oops, sorry, it should start. Okay. And before starting my lecture, so I would like to emphasize what I'm doing is very special. So here you can see the date of the publication, and this has been published just 1 week ago. And person, and it is now registered in some. I'm not sure the people here know about it. PubMed is some database storing papers about biology, and they have functionality to list the similar article to individual articles. Then this paper now is registered in this PubMed, and I have found top 10 similar article selected by PubMed is mine. So this means no other people doing something similar to me. And and the other thoughts, I expect people here, so mainly beginner, in my sense, in the career stage, early career people. And this paper has been rejected 8 journals. And first version of this paper has been uploaded some preprint server. I'm not sure the people here are so familiar with the biological preprint. People here only know about the archive. But we have also some kind of preprint server in biology, and I have submitted this manuscript 2 years ago, and this one does not change at all. So this means, so I'd like to encourage, especially the people who are in early stage of career, even if you have your paper has been rejected many times, don't give up. This paper has been rejected 8 journals, and during 2 years, there's no improvement of paper, but it has been finally published. So in that sense, so perhaps if you listen my lecture, so it is very special and very unique. But I encourage people doing something similar, something no other people are doing. And before discussing about my lecture, so I would like to briefly introduce the genomics, because I expected the people here mainly came from informatics. I'm not sure. So perhaps it is too introductory, but maybe. As I have told you, I applied the tensor decomposition to genomic science, and I would like to briefly discuss about the first genomic science. Genome is one-dimensional array of 4 characters. The audience don't have to understand what this 4 character is. Simply, you can understand from the informatics point of view, the genome is a data information composed of these 4 characters. Then there's a so-called central dogma. Central dogma is dogma in genomic science, and this DNA has been stored almost all cells. Our body also is composed of a huge number of cells, and most of these individual cells have a full set of genome. And of course, the individual cell does not use all of genome, only part. Then DNA, full-length DNA, has been copied to shorter sequence. This is called RNA. And in RNA, not ATGC, but AUGC, character T is replaced with U. And this short copy of long DNA has been then translated to the one-dimensional array of 20 amino acids. And this is protein. Then here is a diagram of how the central dogma works, starting from genome, composed of DNA, is partially copied to RNA and then translated to protein. So this is so-called central dogma, how genome as a digital machine can work. Then this is the translation rule from RNA to protein. As I have told you, RNA is composed of 4 characters, U, C, A, G, and amino acid has 20 kind of proteins, amino acid. Then, in order to express 20 amino acid using 4 characters, we need combinations. The life employed 3 characters to express the 20 amino acid. So, for example, this CGU means first character C and the second character G and the third character U is correspond to this amino acid. So, in this sense, a genome is completely digital machinery to code protein. Protein's function is something like to suppress some function, or sometimes act as sensor, or anyway, protein is real machinery, and DNA, RNA is some kind of data information to store the structure of protein. Then what I'm doing is bioinformatics. So what is bioinformatics? Bioinformatics is some kind of data science. As you know, here, data science is composed of 3 parts: domain knowledge, and statistics, and computer science. And this domain knowledge in bioinformatics is biology. So bioinformatics is a combination of biology and statistics and computer science. And traditional biological science is hypothesis verification. This means we assume something, and in order to check this assumption is true or not. And then, but bioinformatics is data-driven, and we can find some rule from completely data-driven way. So the bioinformatics is some kind of data science which does not start from hypothesis. Then in bioinformatics, what I'm doing? I'm doing is some kind of omics analysis. Omics means some kind of sequence of signal. I will tell you what kind of signal I'm trying to study, but suppose some signal came from the DNA or genome, and purpose is the analysis is to select some important genes. And the reason I would like to select important genes is because, as I have told you, all cells basically has a full set of DNA. But each cell can use only part of DNA. Then it is important to which part of DNA has been used, because dependent upon which part of DNA used, cell is differentiated skin and hair and heart. So this is very important to select which gene is important. And the next problem is how we can select important genes. In my method, well, of course, I'm not the only person using this strategy. People are trying to find genes associated with some contrast. So between experiments. So, for example, we can find which gene is important to skin with compared to hair to select some genes with some strong contrast. And then problem is what kind of contrast we consider. The most popular contrast is amount of RNA. RNA is, I have told you, there's some small part of the DNA in order to translate to protein. And this is some kind of jargon, but we call this amount of RNA transcribed DNA as gene expression. This is some example. Each point is some samples. There are 119 samples came from tumor, and 113 samples came from normals. And this is expression of some genes. So this means if a gene came from tumor, distribution of expression is like this. So this is logarithmic scale. So this means the expression of individual expressed genes in individual samples is so diverse. But in average, it is obvious gene expression of these genes more than normal one. Then in order to check if these genes has different expression between tumor and normals, we compute some p-values. The p-values also is some kind of jargon in statistics. Assume gene expression of these class come from same distribution, and this difference occur occasionally. Then we can compute the p-values this amount of difference occur by chance, and if this p-value is very small, it hardly occur in by chance, then we can regard it, this difference is not accidental but true. And this is, we usually call this statistical test. And here's a p-value. So this means this amount of difference can occur only when, even if these two came from same distribution by this probability. So 2 out of 1,000. And in addition to gene expression, we have many other features to consider. For example, we can consider the DNA methylation. DNA methylation means this is a cytosine. Cytosine is one of the ATGC, C, it's called C. There are 4 characters which, by which the DNA composed of is ATGC, and cytosine is C. And this part of the hydrogen can be replaced with a methyl base. Then, if this is the genome, and this part is gene which is translated to the protein, and if some part of this location of DNA is methylized like this, this translation will be disturbed. So instead of observing gene expression directly, we can also check this gene is functional in some cells, considering if this part is methylated or not. Or alternatively, we can consider the histone modification. The histone is some kind of protein around which DNA wrapped. So DNA is lengthy string without organized DNA in some structure. It is elongated and it's very difficult to make use of. Then DNA wrapped around histone. This is the histone is this part is one histone, and this cylinder is composed of 4 histone, and DNA wrapping around this histone. And if DNA wrapped around the histone very tightly, this part of DNA cannot be translated to protein. But if this wrapping is loose and this part is bared, this part can be translated. So then instead of the measuring methylation or gene expression directly, we can also measure this part is bared or not. And if this part is bared or not, is affected by the histone modification. This is small particle, so molecular bind to the histone. If some molecule bind to histone, the wrapping is loose and this part can be translated, otherwise not. Then we can measure if this part is modified or not. Then we can measure difference. So we can make use of the amount of histone modification to find if there's a contrast between two conditions too. And of course, there are many kinds of contrasts, but I have no time to explain all of them. So instead of the measuring a histone modification, of course, we can directly measure which part is bared or not. Here, like this. And without measuring the gene expression and the DNA methylation and the histone modification, we can also directly measure which part of DNA is bared to know which this part can be functional. And also there are many kinds of contrast we can consider, but at least 4. But the main point of this talk is not what kind of contrast we consider, and I would like to discuss about how we can find the contrast. Then, of course, I will spend half 1 hour to discuss how we can find the gene which is contrast between two conditions. So it should be difficult, otherwise I don't have to study it. Why it is very difficult? This is mainly because this is so-called large p small n problem. Large P small n problem is a famous problem, so people here of course know. And this means we have only small number of samples with many number of variables, like this. So if we express this structure is as a matrix form, the small number of samples, but each sample is associated with too many features. Why it is difficult? For example, we would like to discriminate tumor from normal people, and this is labeling to express individual person have a normal tissue or tumor. Each line represent each person, the n person. And A11, A12, A1n is gene expression of n genes, first person. And this is gene expression of n gene of second person. And we have selected n genes among the huge number of p genes. Then, as you know, we have always have a solution in this set of equations. So this means we can use any kind of set of n genes among p genes. So this means we cannot use if n genes can predict the individual person is tumor or not in order to find which genes are important, because any set of n genes can predict completely. So because of that, if the number of variables here, the p, here's the number of genes, much larger than the number of samples, it is important. It is impossible for us to find which genes is important in order to check if this set of genes can predict outcome. Because if we have same number of genes with the number of samples, we can always have a solution. Pardon me? This is, okay, sorry. A11, A12, A1n are the n genes for first person. Yeah, B1 is some label to predict this guy have no. P? Yeah, P. We have selected arbitrary n genes among P genes. For example, there are 1,000 genes and the number of people 10. We can select any 10 genes to predict. Yeah. Then we can find any which genes is important, because any set of n genes can predict if this guy has normal tissue or tumor completely. Okay? So because of that, if the problem is large P and small n problem, it will be difficult to find which is important. So another difficulty came from the statistics. So as I have told you, if some gene has this contrast between tumor and normal, this gene should be important, because the probability this can happen by chance is very small. But suppose if we have 1,000 genes, then even if each contrast can happen very small amount of p values, if we have 1,000 genes, the expected number of genes, this contrast by chance, is 2.6. So this means even by chance, we can find 2 genes which is important because it has contrast between tumor and normals. Then even if we have very small number of p-value, we cannot say this gene is important. And much worse than that, here we assume number of samples, number of patients, number of subjects is small. And I cannot explain it explicitly, but it is obvious. Even if these two have the same contrast, the number of samples is small, it can happen by chance. But it is hardly happen by chance. If we have 6 samples, and it can easily deviate 3 low number, 3 higher number by chance, But we have this number of samples, here 18 samples. It is hardly deviated between two classes by chance. Then if we have large p small problem, because the small number of n, the probability this can happen is large, and the number of p is large. So actually speaking, the number of genes which can have some contrast by chance is probability. So the upper case is probability, and the smaller case is number of genes. Sorry, so confusing, but this is by convention, so I dislike to change it. Small p is the number of genes, the large p is the probability it can happen. And small p is large, and large p has tendency to large because of small number n. So this is very bad. We have many number of genes which can have contrast by chance. Actually speaking, this problem is general. There's no unique solution to solve it. But we can propose some domain-specific method. Then in this talk, I would like to propose use usage of principal component analysis and tensor decomposition. And people here came from the informatics, perhaps you know PCA and tensor decomposition well. But I would like to briefly explain what the principal component is and tensor decomposition is. This is, suppose this Xij is a data composed n samples, m samples and n genes. Then this matrix mathematically decompose product of n times L matrix, and this columnar vector is usually have normalized. This means mean 0 and standard deviation 1. Then we have to have some diagonal matrix which expresses the weight of product. And from right hand side we have L times M matrix. This multiplication can result in this matrix. This is PCA. And mathematically we can write this equation like this. And as I have told you, this is n is the number of genes and m is the number of samples. Then how we can make use of this procedure to select genes? And VLJ is vector attributed to the samples. And suppose we have samples composed of the healthy control patient. And if some L, this vector is distinct between healthy control patients, then corresponding vector U of this L, the gene expression looks like this. Then this part has larger contribution, so this means gene expression of individual genes having this amount of value is consistent with this. Then we can regard it, this can be important genes. If the opposite sign is associated with this part of genes, we can expect reversed contrast. So healthy controls has less expression than patients. And this is the way how we can make use of PCA to select genes. Later, I will tell you why it works well. It works better than the conventional method by synthetic data, but at the moment, please trust me. And this is a brief demonstration how it works. So suppose we have n samples and n genes, m samples. Only small number of genes has distinct between 4 classes. Then we apply the principal component analysis to this and embedding this each circle corresponds to each gene. And if we use first two vector attributed genes, we can place the genes into the space. And some genes are apart from the origin, and if this direction corresponding sample vector is distinct between 4 classes, we can expect these genes correspond here, which are distinct between 4 classes. So this is a synthetic example of how it works. Suppose we have 100 genes and 20 samples. 20 samples are divided to class. Each class draw from Gaussian distribution, having same standard deviation, but one has positive mean and others has 0 mean. Then we apply PCA and use first vector attribute genes in order to express each point represent this one of 100 features. Then this part, this red triangle corresponds this part, and blue cross is a top 10 feature. Here, this is the origin. Top 10 feature apart from origin is cross. Then as you can see, 9 out of 10 outliers coincident with 10 features distinct between 2 classes. So using this method, we can select which feature is distinct between 2 classes. The important things, we don't need to know anything about how many features are distinct, and how many samples there, and which one belongs to which class. This is completely unsupervised method. In spite of that, we can select number of features which are distinct between 2 classes. Of course, it depends on the standard deviation, the mean values. If the mean values is smaller, of course, the distinction is very vast. And next, I would like to demonstrate how we can extend to tensor decomposition. In tensor, we have 3 suffixes instead of 2. In the previous case, we have n is the number of genes, m is the number of samples, but in addition to this, we have suffix k, which represents the number of tissues. Then the xijk represent the gene expression of ith gene, which belong to js subject, and in case tissue. I don't have enough time to explain it. In some mathematical procedure, we can also decompose into the product of vector attribute only genes and samples and tissues. And I make use of a higher order singular decomposition, higher value method. And anyway, so the point is we can have decompose this tensor into the product of 3 vectors, in each of which attribute only gene and samples and tissues. And how we can make use of this expression to select genes. So suppose some specific L2, which is attribute to samples, also distinct between patients and healthy controls, as I have done in PCA application. And in addition to this time we have the suffix to specific to tissue. We may have this kind of vector, which is only have large values on some specific tissue. Then this to represent the tissue-specific distinction between healthy controls of patients. Then you fixing L2 and L3 and G is weight. And we would like to know which L1 has larger contribution to combination to L2, L3. Then suppose this vector has larger G with fixed L2, L3. We can again find, for example, this part. This part is opposite sign. Then tissue specific differential express gene. So previously we have find some genes distinct between healthy control and patients. But in addition to this, now we can have tissue specific differential express genes because we have another suffix here. Okay. So any questions from to this point? It's okay. Pardon me? Oh. What do you mean? So, you mean you don't know how exactly select it mathematically? Okay. So, basically, we don't find which is larger contribution compared to.
Each line corresponds to one gene, right?
Yes, yes. Each bar corresponds to each gene.
So some genes are overexpressed and some are
Down. Yeah. Yes.
underexpressed. And then,
That you would like the mathematical solution. Okay. So I will explain it later. And okay, so he has asked how we can select this part mathematically. So I will tell you. So this is some synthetic examples. This example composed of the 1,000 variables. Each of variables are associated with 36 samples. 36 samples compose the 6 experiments times 6 experiments. So each experiment has two distinct between two classes. So for J, if J less than 3, Xij draw from Gaussian distribution with non-positive mean. Otherwise, which we draw from 0 mean. And then the situation like this. Only when the K and J is less than 3, this part is drawn from Gaussian distribution having non-zero mean, otherwise 0. And this can happen only first 10 features. And for other features, Xij, all drawn from Gaussian distribution having 0 mean. The task is to find which genes which genes are associated with this description. Simply, we apply tensor decomposition. J and K is vector attribute samples. The first vector shows some distinction between two classes. J is less than 3 and J is larger 1. And some vector attributed, some vector attribute to the K. First vector attribute to K, distinction between K is larger, less than 3 and K is larger 3. Then we have noticed L2 should be 1, L3 should be 1. Then by investigating the G, we can find L1 equal 1 has largest absolute variable G. Then the G looks like this. Then it is obvious this part has 3 dimension G. G is L1. UL1I is a vector attribute gene, and L1 is a suffix of the vector. We have many 1,000 number of vectors. The L2 from 1 to 6, it is equal to number of samples, and J attribute J samples, L3 K attribute K samples. Is it okay? Yeah. It is obvious this is outlier. Pardon me? You mean a long experiment? Yeah, of course, if you expect it long, there's no way for us to check it. It is completely data-driven. Okay, then as I have promised you, I will now compare standard statistical test. We apply standard statistical test for this problem. Only one of 10 features supposed to be distinct have small enough p-values if we consider 1/3 of genes. This one. So fails rate 90%. So it is completely useless. But using tensor decomposition, it is very obvious this 10 is outlier. Now this means this kind of method is very useful if number of samples is much less than number of features. practically. And of course, in this case, it is obvious this part out here. But practically, these outliers are not separated clearly from others. Then we need some mathematical formulation to attribute P. This P value represent outlier can happen by chance. Then we assume this vector attribute G composed of the Gaussian part and outlier part. And if we attribute P values, assuming a Gaussian distribution, this part have flat histogram of 1 minus P, and outlier here has very small P values. So by tradition, we plot 1 minus P horizontally. The small P has peak at 1 minus P equal 1. So we can select this part, assuming UH1I obeys the composed of the Gaussian distribution and outliers. Then we try to optimize sigma U, which represent Gaussian distribution of null hypothesis. We have changed sigma U in various numbers. And as I have told you, if ULI composed of Gaussian distribution and outliers, and there is some peak here, and excluding this peak, this part should be flat. Then I compute the standard deviation of this histogram and select best sigma U having smallest sigma. Sigma H is the standard deviation of this histogram. This should be, since this part should be flat, smallest sigma H should be associated with the best sigma U. And we select this sigma U, because if we change sigma U and we plot sigma H, this is the standard deviation of histogram, this is the standard deviation of the Gaussian null hypothesis. This is the smallest values. Then employing this sigma U, and we compute P values and attribute to the 1,000 features, we can successfully find 9 out of 10 correct one, even if we multiply 1,000. So this method is much better. Only when the number of P, number of features are very large compared to the number of samples. And statistical test fails to find correct number of genes. In addition to this, statistical test has another problem. In my previous synthetic example, we try 6 times 6 equal 36 samples. If we increase number of samples to 10 times 10 to 100, then this time all of these features are correctly selected by statistical test. So this means, depending upon the number of samples, we can have different answer. If we have very small number of samples, this set, there is only one feature distinct between two classes. If we have much larger samples, this statistical set, there are tensor. So it is not very good, because what we are doing in natural science, depending on samples, if outcome is different, it is very bad. But PCA, we have already select 9 correct genes, and if we increase the 10, this number does not increase, only we can select 10 features. And this is very important feature, as you have noticed. This strategy can be applicable all cases, not only restricted to the biology. But I don't know why the people did not discuss about very precisely this point. And then I have written one book, second last year or third last year, to discuss this problem generally. So if we are interested in this kind of problem, how we can use tensor decomposition and PCA to select features under the large P small problem. Please read my book. So, any questions here? Okay. Then I think we have 40 minutes. Then I would like to apply this method to real examples. The first example is gene expression. And gene expression is, as I've told you, the popular topics to find important genes by investigating a contrast of gene expression between conditions, for example, healthy control and disease patients. And I have write one preprint this year, and this was just accepted. But as I have told you, this paper, I have write this preprint past this February, and this preprint just accepted 1 week ago, and it took 7 months to be accepted. And just like the paper I have introduced in the beginning lecture, which took 2 years to be accepted without any improvement. This paper is also simply took 7 months to be accepted because no other people can review this paper because I'm the only person to do these topics. And in this paper, I have applied my method, PCA1. And I have no time to explain what the RNA-seq technology, but please understand XIJ, using this technology, we can find XIJ, which represents the expression of ice genes of J sample. And as I've told you, the problem I'm doing is large P small n problem. Now we can tell you what kind of large P small problem we have to discuss. This is a benchmark data set. So it is very usual for this field. So number of samples, only 14. This is, half of them are composed of some of the universal human reference. This means this is mixture of all tissues of human. So there's no tissue-specific expression at all. And compared one is human brain reference. And this is composed 7 and 7. And this is only 40. But the number of genes is this number. So it is a terrible large P problem. And it is very usual for this field. And this is not an synthetic example. So you have to understand what kind of data set it is. So this is standard visualization that people in the field would like to use. Horizontally, we have to take mean logarithm mean values. Each point represents each gene. Each gene associated with 40 samples. Then if we take average, each gene has average expression, and they would like to take logarithmic values. Horizontally, they have taken contrast of mean within class. So this is the mean value of 7 human reference samples and human brain samples, and divide each other, and then take log. So this is logarithmic values of mean value and ratio of mean value within each class. And distribution, this is distribution. So this line is 0. So logarithm value 0 means ratio between two class is 1. So this line represents genes does not have a contrast between two classes. The majority of genes are concentrated the place where gene does not express any contrast between two classes. Then people would like to find small number of genes which are distinct between two classes. So as you can see, this is terrible problem. But if we apply PCA, simply we can find this. So this is the first vector attributed, this is the first vector attributed genes, and this is second vector attributed genes. Then corresponding sample vector, first vector attributed genes, and first vector attributed gene, the second vector attributed genes, like this. Here. First one is simple summation of 14 samples. This corresponds to mean. The vertical axis, second vector attributed samples, is opposite sign dependent on the classes. So this is completely unsupervised. I don't assume anything, but data automatically give us mean and deviation. This reminds us standard conventional plot. They have vertically taken the logarithm ratio of two samples, and horizontally they have taken the mean value. So without assuming anything, we can have qualitatively same plot. But the distribution of genes is completely different. So as you can see, distribution of genes is very different from conventional plot. And this is the distribution of density of the genes. So as you can see, almost all genes are concentrated here. This is no expression and no difference. So this is the reason why the people traditionally would like to take logarithm value, because if they don't take logarithm value, outcome like this, so there's no distinction. But this is more reasonable plot, because it is obvious what part is outlier. The purpose is to find which part is outlier. So it is obvious this gray part is outlier because almost all genes are concentrated here. So clearly, PCA is much better than a much better way to represent these genes to find outliers. Then, as I have told you, PCA can have more than two components, but in this case, we have only two components. This is the contribution of each component, and the third component larger than 3 has no contribution. So this is essentially two-dimensional embedding. So it is not reasonable to embed 40,000 genes into two-dimensional space and find outliers using this plot. Okay, and more important part, this PCA plot can solve some problem which exists in conventional method. The people here are not came from the biologists, but it is also easy to understand. But genes with high expression should be more selected. So this means if we discuss out the people from biology, they think genes having more expression has more importance because life has spent more power to express this gene. But as you can see, in the standard visualization, horizontal axis represents the importance of gene from the point of biology. But as it is obvious, genes having larger expression has less distinct between two classes. So the genes having contrast between class has less expression. So it is very controversial. Then people assumed some artificial constraint here. Mu is the mean value, and sigma u is standard deviation. So they consider u in some values, and this is correspond to vertical axis, vertical line. Then along this line, each set has some Gaussian distribution having this mu. Then they assume artificially this Gaussian distribution should have smaller standard deviation if mean value is large. To my knowledge, there's no biological or mathematical basis to assume this. But without assume this, this part can never be selected because clearly expression, genes having larger expression has less expressed between two classes. Then they assume this. Then using combination with this assumption with some statistical, this is a state-of-art method, statistical method to select genes based on the p-value. They can find this part can select because of this assumption. But as I have told you, using PCA, larger expression associated with larger distinction. So we don't have to assume this. Simply we can select genes having larger distinction between two classes in PCA plane. We can find this plot because larger genes having larger value expression is much frequently selected because of this embedding here. So large genes having larger expression has large deviations. Then without assume this kind of empirical equation, we can find reasonable selection of genes which satisfy the requirement. Genes having more expression should be selected more. So the reason why in log plot we have this kind of distribution is very simple. So we have selected genes by deviation, not logarithm ratio. Then this plot is log plot. If we plot genes selected with deviation by logarithm scale, we have this kind of expression. This is threshold value based upon deviation, and this is mean this expression. It is obvious if large expression associated with the mean values, threshold LFC can be smaller like this. But this equation has been introduced by Manhan, but this formula came from by data science. So I think the reason why they have to introduce this kind of empirical equation because they did not notice the PCA is better representation of data set, and they wrongly assume the log plot. Then they have to introduce this kind of empirical equation. But if we can stand for data-driven science, it is natural represent genes on PCA because horizontally you can have mean value and vertically you have distinction, and it is definitely two-dimensional plot. And next, we would like to optimize sigma u as I have done in synthetic examples. So we attribute p-values to genes simply assuming the Gaussian distribution and standard deviation drawn from using all samples. This is a histogram H, histogram 1 minus p. It is very different from the ideal case. Ideal case is the constant value and peak here. It is very different. Then we try to optimize which part of genes are peak and which part is flat. Of course, it is not very good, but it has some additional peaks. But if we optimize the sigma u having a flat distribution, exclude the peak, we can have some optimized sigma u. And using this optimum sigma u, we can assume Gaussian distribution at the p-values. And p-values will be collected considering the number of genes, and we can select genes. Then the biological meaning of the select genes is very good. So this is comparison with DSX2, which is conventional statistical test to select genes which are distinct between classes. And this is evaluation of genes compared with a known set of genes having biological functions. This is not my method. It is very usual method. If we select some genes, each set of genes compared with if genes associated with some biological functions. If all of selected genes are associated with these biological functions, and genes not selected are not associated with biological functions, this set of genes is associated with this biological function. Then we compute p-values. This distribution can occur by chance, and if p-value is very small, we can say that this selection of genes, not by chance, associated with this biological function. This is a tissue-specific set of genes drawn from data set, and the white bar is dissect, which is state-of-art method. And of course, dissect is very good. So this is logarithm p-value base of 10. So this is 10 to the 50 or 70 or something like that. So this dissect itself is very good. But even compared with this, our method is much better than that. Actually speaking, I can plot exact p-values for these categories because it is too high. Even comparing with the state-of-art method, dissect, very simple PCA can select genes more consistent with tissue specificities. And I have no time to explain these tissue specificities. These are all related to the brain. You should remember this is the contrast between brain tissue and normal tissue. And of course, I have no time to explain detail. There's many other state-of method, AGR, and NAIST, and Boom. And even comparing with this method, this is other biological functions, and logarithmically you have taken negative sign, the logarithm p-bar is base 10. So always it outperform other method. So this means the PCA, in spite of the very simpleness PCA, it can outperform, from the biological point of view, many complicated state-of-art method. So the conclusion of the application to the example is this. The PCA can outperform various state-of method, statistical test, and PCA does not have to assume empirical equation. The threshold value should be less for larger expression. It is automatically satisfied. And second, selected genes much more consistent with biological values. So personally, I think state-of-art method is wrong, and people should process gene expression using this kind of technology. But I don't think people agree with me because this method has been used over 10 years. It is too late to change the strategy. So any questions here? Okay. Okay. So we have 25 minutes. So I will briefly discuss the application of other method. So second application is more realistic. So I have applied this method to drug repositioning for COVID-19. And I think the people here mainly came from informatics, so I would like to briefly explain what the drug repositioning. As you know, the situation COVID-19 is much better than 22 years ago because of vaccination. But we don't have some problem, still have, so we wear masks. Of course, I know other countries do not wear masks anymore, but anyway, number of people who have infected with COVID-19 is still large. The reason we could not completely overcome the crisis of COVID-19 because we don't have any effective drug. But developing drug is sometimes very time-consuming process. And for example, even if we can find some compound which is effective to COVID-19, in order to take through mouth, this should be water soluble, and it does not have any side effect. And there are many requirements, even if some compounds can suppress the COVID-19 amplification. But if we can make use of a known drug for COVID-19, it is more effective because the drug has already been approved for human use. And if some drug is turned out to be effective with COVID-19, we don't worry about the water solubility or side effect or not. Then drug replenishing is one of the good way to find good drug for COVID-19. And the strategy to find drug for COVID-19 with a method is like this. First, I downloaded some gene expression from this database, and this composes this number of genes. This is the P number of features, and the experiment repeated 5 cell line. This is not true human tissue. This is cell line incubated cells in Petri dish. Then the comparison that this is mock virus, which does not have functionality COVID-19. This is COVID-19 virus SARS-CoV-2. Then this process repeated 3 times. Then we have 13 samples for each gene. The 5 is number of cell line. The 2 is distinction between infection and non-infection. The 3 is the number of replicate. Then we have 30 samples for each gene. Then this is terrible large p small problem because number of features 10 to 4th, but the number of samples only 30. And this is terrible large p small problem. So it is worthwhile trying apply our method to this data set. Then what we will find is like this. This gene expression can be represented by the fourth order matrix. This is the number of genes. This is P, and this is number of tissue. The cell line. This is distinction between infection and non-infection. This is the number of replicates, and this XIJK tensor can be decomposed of the product of the vector attribute, the cell line, the attribute, the distinction between infection and non-infection, and 3 replicate, and the gene. And considering these vectors, attribute the cell line and distinction between 2 conditions, infection and non-infection, and replicates, we will find some genes whose expression independent of the cell line and replicate. So this vector attribute to the cell line and replicate should have constant value. But this one, the attribute distinction between the infection and non-infection should have opposite sign. And as you can see, we can have a vector satisfying these requirements. First vector attribute to 5 cell line has almost constant value. So this means gene expression independent of cell line. And second vector attribute to the distinction between infection and non-infection has opposite sign. And first vector attribute by the replicate has constant value. So we fix L1 equal 1, L2 equal 2, and L3 equal 1, and which L4 attributed gene have larger G with fixed L2 equal 1, L3 equal 2, and L4 equal 1. Then we can find fixed one have the largest G. Then this means we can make use of fixed vector attribute gene to select important genes. Then I don't repeat the procedure, but optimizing standard deviation such that histogram of 1 minus p composed of flat part and peak, we can find this number of genes with corrected p-values less than 0.1. Here, this is histogram 1 minus p without optimization. This is optimized one. And we assume this histogram 1 minus p, and optimized standard deviation is this value. Then, and we have successfully find this number of genes. So this number of genes, actually speaking, reasonable. If we use other method, the decision is terrible. So this is a standard test which is used not only biology but general statistics t-test. There's no way to apply this test for all of 5 cell lines at once, so we have to apply this test to each of cell lines one by one. Some case we don't have any gene judged as expressed between infection and non-infection. For this cell line, only 1 genes, but for this case we have 325 genes. So it is very inconsistent. It should not occur because we have 5 cell lines, but this is not so different. It is clearly very odd result. So it is unlikely there's no genes different express infection and infection for this cell line, but there are 300 genes which are different expressed between infection and infection for this cell line. It's very odd. So biologically acceptable. For this is some kind of t-test, but I have no time to explain. This is adaptive t-test to be used for gene expression analysis. It is completely failed genes which is expressed between 2 classes. And Lima is some kind of more advanced method. And this time, it has selected this number of genes. So it's not bad. But actually speaking, as you can see, total number of genes is this. They have ignored most of genes to have this result. And so, if Lima has special skill to exclude genes before treatment, then they have a reasonable number of genes, but without this exclusion, Lima cannot work. And these X2 I have used in the previous example has similar problem. So number of genes selected fluctuate from sample to sample. So, and then I would like to find drugs which is effective to COVID-19 based on method. So this has been 2 years ago, so we don't have many investigation COVID-19. But this is a comparison with human genes, which is known to contact with SARS-CoV-2 protein. Each line represents a SARS-CoV-2 protein, and this is a p-values, which is known to contact with these genes or within the human protein. These p-values how selected genes are coincident with human genes which is known to contact with this SARS-CoV protein. And this one, black bar is best method we have used. Again, the coincident with very good, and de facto standard DSX2 is here, so it's very bad, as you can see. So here, so this is the DSX2 and this is ours. This is the Lagrange p-values, base 10. So again, from the biological point of view, our method is much better than the state of method DSX2. We have more confident human gene, which is known to make contact with SARS-CoV protein. Then we try to find drugs to be effective to COVID-19. And the way to find drugs which affect COVID-19 using genes we have selected is very simple. There are some database for drug compounds, treatment of each compound affect some genes. And in database, we can find which genes expression are affected the treatment of compounds. So, for example, this means there is 442 genes whose expression altered by the treatment of these genes, this drug, and this number is overlap of genes with selected genes by us, 3,000 genes. And this is the p-values. So again, this is very small, and we have many drugs whose affected genes are coincident with our selected genes. And this is genes whose expression down treatment of these drugs, and this is genes whose expression down because of treatment of these drugs. And I don't have enough time to explain this, but these drugs are already been tested to COVID-19. Almost all of them has once tested to be COVID-19 drugs. So what we have done very simple. We have analyzed gene expression by tensor decomposition and select genes and select drugs which affect these selected genes. These simple procedure can rank drug, and almost all of them are drugs which has already been tested to COVID-19. So this means our method is should be very effective to select some drug for some diseases. So it procedure should be applicable to any kind of diseases, not only to COVID-19. Yeah. So, any other questions? Sorry, just too specific. Maybe. So I have only 10 minutes, and so I have no time to explain this very precisely. And third example, so we have also applied the DNA methylation, and we have written preprint. And this one accepted some conference and will be published in some journal tract. And I don't have enough time to explain the detail of this result. But even if we can apply the same method to methylation, so as you can see, the similar. So this is constant for 6 samples and distinct between 2 samples. This is the 4 sample cases, and this is constant and distinct between 4 samples. And we can have this kind of optimized histogram of 1 minus p, and we can select methylation. And we have compared with some state-of method, and ours much better than coincident with known DNA methylation part in database. So it is also completely data-driven. And sorry, I have no time to it. And we have also applied histone modification too. And this is p-value. This is this histogram 1 minus p are a little bit odd, but again, our method can select histone modification which is coincident with known database. So, sorry, I could not find enough time to explain histone modification of DNA methylation, but perhaps you are bored. And take-home message is like this. And PCA and TD can at home various state-of method. I don't have enough time to explain the method used in DNA methylation and histone modification. But many people try to develop method to find differently methylated genes, different genes associated with different histone modification to several algorithms. But without modified details of the procedure, simple PCA and tensor decomposition can outperform as a method, independent of the we consider gene expression, the DNA methylation, the histone modification. So the reason that this method works very well is simple. So if we don't assume anything and simply we consider the data set and let them speak, they can honestly tell us what's true. If you can deal with your data set as much as possible, and if you let them to be as it is, you can find much more biologically true futures. So I'm not sure, because people here mainly not came from biology, but it can be true in any other fields. So because of the popularity in machine learning, very complicated and supervised method is popular, and because of that my paper has been delayed to be published. No one interested in this kind of unsupervised method. But I think if we can make use of this kind of unsupervised method, this method can outperform conventional standard method. The reason the unsupervised method is not popular because simply they do not try. Because it is something odd. It is better to find futures without considering label. It sounds like something controversial. We should make use of any knowledge we know, but the answer is no. So we don't have to make use of knowledge prior to select futures. The knowledge about futures should be used only validation, as I have told you. And when selecting the future, we don't have to consider the labeling, and it can be much better if we can deal with data set correctly. So, okay. So this is what I would like to tell you. So thank you very much for interest.
Thank you very much, Professor Taguchi. So we have a discussion. So if you have any questions, ask on site by mic and or also online.
But if sometimes data is not so much good, I think. For example, you saw an example where we found that PCA, first two components correspond to more than 90% variance. But if it doesn't happen for some data, can we apply also?
Okay, question is if the contribution of first or second component is not large, so it is effective or not.
Yes.
So actually speaking, the answer is yes, because I previously, as you can see, so for the application COVID-19, we have used fifth component. You don't think fixed component has much contribution. It has very small amount of contribution because it is fixed one, but it is very successful. This means even if the component we consider does not have large contribution, it is effective. Because the number of genes selected, 3,000, is very small amount of the part of the total future. If we select only 10% of future, of course contribution cannot be large. So this means in order to make use of our method, the component we consider does not have to be large contribution. Even if contribution is small, it simply means the future is distinct between condition is small. Okay?
Maybe makes sense.
You can trust me.
Okay, may I have a question? So, in my understanding, your model based on PCA, so it's calculate the variance. So it's supposed you assume the original data noise on the original data should be some kind of the regular or Gaussian type. But when we observe the gene expression of RNA-seq data, the distribution of the expression is not so always the normal. So do you have any idea about such application to extend the?
No. If you applied principal component analysis, it obeys Gaussian. You say low value. low value distribution of gene expression does not Gaussian.
Mm-hmm.
But if you take PCA, here, distribution U2I is well described with combination of the peak and here. This part is usually discarded in DSEC2 because DSEC2 has procedure to exclude the genes whose expression is very low. Then this part cannot appear in DSEC2. But our method does not have to do this. Simply, we can require flat part and peak as much as possible. So low value, you are right. Low value does not look like Gaussian. But once we take tensor decomposition of PCA, it's much closer to this Gaussian.
Any questions? Okay, I have.
Okay, thank you.
So we are also trying to solve the same question of the large number of features and small number of samples. But not so large fraction. I mean, so in your case, the samples number is so small.
Yes.
So in that case, I am afraid that the small number is easily biased. So that the bias sometimes may hide the important features or sometimes make difference between the features. So do you have any idea about the bias
Okay.
problem?
Okay. The question is if because of a small number of samples, it is easily affected the noise because noise is large. In my opinion, this problem has been avoided by the considering, oops, not this here. So as I have told you, in the case of the application of the COVID-19, we have used the fifth component here. And I think I did not count, but if I compute the contribution of this part, it should be only a few percentages. So the answer to your question is noise part should be contribute to this part. Because noise, as this fifth component is coincident with this expression, constant for cell line, the constant replication, but distinct to samples. So if there's a noise, it does not have any projection for this space because it is noise. Taking a tensor decomposition, we can consider this part, which is orthogonal to any noise. Because noise is random number, if we take projection to this space, it should have zero contribution. Then we can automatically exclude any noise. This is my opinion.
So, any question? Other question? Okay.
じゃあなんか、オンにしてもらって喋ってもらえば。
もし上里さん、もしチャットに書けるようでしたら。挙手されてますが。
すみません、上里さんがもし質問があったらチャットかマイクをオンにして喋ってもらって構わないんですけど、聞こえますか？なんかもう終わっちゃった、帰っちゃったかな。もう時間過ぎちゃったから。
ログイン
そうですね、はい。まあでも、もう授業時間一応終わったので。
佐藤さん。あ、ありがとう。
なんて言ってます？こっちにはマイク、音が入んないのね。上里さん、もう一回喋ってくれますか？すいません。聞こえなかったみたい。
はい、すいません。発表ありがとうございます。
どうも。あれ？
正直理解があんまり及んでないので、質問ができるっていう感じがしないんですが。
はい。どうぞ、質問してください。
どこについてきたんですかね。すいません。実験3のとこで、基準手法が3じゃないか、実験2かが、学リポジショニングの話で、基準手法を一つするっていう話があったじゃないですか。あれ、またお風呂聞こえてないですか？
聞こえない？
やっぱりチャットに書きます。はい、
すいません、チャットに書いてくれるそうです。ごめんなさい。すいません、もう時間は終わったので、皆さん帰ってくださっても結構です。ごめんなさい。授業時間終わったので、退出だって結構です。すいません。今、チャットで質問を受けてから僕は答えますけど、皆さんどうぞお帰りください。すいません。伸ばしちゃって。チャットで質問書いてる？これあれだね、質問するの難しいんだね。なんかでも何も来てないね、チャットで。チャットになんかまだ何も入っていないみたいですね。そうですね。
This class is over, so you can ask after the satsang, and no questions, so you can leave anytime.
あ、これです。
講義前にお知らせするのを忘れたんですけど、先生も
どうぞ
お待ちしていたので、もしよろしければこちら。
ありがとうございます。 ありがとうございます。はい、ありがとうございます。
発表ありがとうございました。ドラッグリポリションの例について、T テスターもリマとフューチャーセレクションの結果を比較する部分があったと思いますが、リマの方をうまく適用できる比較対象となる基本子がほぼこれはそもそも有用な既存子がないためか、それとも十分なセレクションと考えてよいのか、確認させてください。質問、このドラッグリポジショニングについて既存手法との比較があったっていうことなんですけど、基本的には、うまくいってないということで、一番よろしくないのは、DSEC2 っていうのが一番世の中で信用されている方法なんですが、これは実は僕がやったわけじゃなくて、このセルに投稿された元論文のアペンディクスにこういう結果がすでに載っていてですね、DSEC2 でやるとセルラインごとに、結果がめちゃくちゃになっちゃってるっていうことはもう書かれていて、で、彼らは結局、セルに乗るぐらいだから、この327項が正しくって他は間違っているっていうことで解析を進めてるんですけど、それは、僕は、嘘だと思っていて、全然うまくいってないのに、一個だけうまくいったと言っていると、私は思っています。なので、ご質問の、他の方法がうまくいってなかったことについては、僕は多分、全部ダメなんだと思うんですけど、こういうのは実験を、やっぱり COVID-19 最初にやったっていうことが評価されて、論文になってしまったのかなと、私は個人的には思っていますけど、なのでお答えとしては、統計的な解析に関しては、全くうまくいってないんだと私は思ってます。これ先ほど私、サンプル30個で、2万次元って言ったんですけど、セルライン5個に分けてしまったら、実は、30÷5 で、6サンプルしかないんですよね。で、6サンプルかける、2万とかでやってるから、さらに厳しくなってるんで、まともな結果を出すのはそもそも無理なんだというのは私の理解です。これでお答えになってるでしょうか。はい。
じゃあこの手法が実上最初の有効な選択の手法であるということですね。はい。
はい、そうだと。私はだから他の方はダメでこれでやらなきゃダメなんだろうなと統計的にはこの方以外ダメだと思っています。ありがとうございました。もし他に質問がなければこれで講義を終わりにしたいと思いますので、ウェブックス入っている方も退出してくださって結構です。今日はどうもありがとうございました。